Last thing to talk about is the nucleic acid. From its name, nucleic, because it is found inside the nucleus. Yes, and it's found in the nucleus of every cell. There are a number of long strands or threads that are known as chromosomes. Chromosomes are equivalent of DNA. Till 1953, we don't know that the chromosomes are DNAs. Before this, we named them as chromosomes. Chromo in Latin means colored, by the way, colored. Zome means bodies. So in the past 200 years, we stain the cells and we can see them if they are during the metaphase, specifically in the division. We saw a color bodies. We don't know their chemical structure until James Watson and Francis Crick came in 1953, and they discovered that its chemical structure is deoxyribonucleic acid, or DNA. So whenever you listen to the word chromosome, it is the DNA, no difference. Okay. What is the basic structure? This is the most important thing I need you to focus on. It's made up of, or what is the basic unit? The basic unit is nucleotide. It's called nucleotide. What is the structure of the nucleotide? Three basic components. Number one, a sugar. Number two, a phosphate group. Number three, a nitrogen containing base. Again, a nucleotide, we have sugar, we have phosphate group, we have base. And we can classify the genetic material into DNA and RNA. It's very important. I don't need it actually now, but I need you to know it later in the inheritance. We have DNA, we have RNA. I'll use this for a comparison, and our first session for the introduction of bio will be over. In the DNA, what is the difference between DNA and RNA? First is the sugar, okay, the sugar. In case of DNA, it is deoxyribonucleic or deoxyribose, deoxyribose. Okay, let's make this clear. D means elimination or removal. Oxy, so removal of oxygen. Okay, ribose. Ribo is a 5 carbon. O's mean sugar, so 5 carbon sugar. RNA has also 5 carbon sugar, but simply it is ribose. Ribose is pentose. Pentose. Penta means 5, O's mean sugar. So it is pentose. Okay. So the first difference is the type of the sugar. Second is the double helix. Okay. The DNA is mostly double strand. While RNA is single strand, mostly single strand. And the base, the nitrogenous base. In case of DNA, we have adenine that will be paired up by a double hydrogen bond with thymine as a nitrogen containing base, and guanine will be paired by a triple hydrogen bond with cytosine. While in RNA, the adenine will pair up by a double hydrogen bond, not to thymine, but instead to uracil. And guanine, same as DNA, will be paired by triple hydrogen bond to cytosine. Those are the major three differences between DNA and RNA. DNA has a deoxyribose sugar. That's why it is known as deoxyribonucleic acid. And RNA has ribose sugar. That's why it's known as ribonucleic acid. Then the strand itself, in case of DNA, it is double strand. In case of RNA, mostly it is single strand. The base pairing rule, actually the bases, the scientist that discovered this was a German scientist known as Erwin Chargaff, and he has the Erwin Chargaff base pairing rule, or you can abbreviate it by saying base pairing rule, in which the adenine as a nitrogen-containing base is always pairing with thiamine by a double hydrogen bond, and guanine is always pairing with cytosine by a triple hydrogen bond. In the RNA, in contrast, adenine pair with uracil, and guanine, same as DNA, pairs with cytosine.